Okay, yeah, we'll be starting now. So, welcome to the course. This is week one of Biological Science Research Program, and I'm the organizer. So, let's get started. So, welcome to the course. This will be a 4-week program. The course is intended to teach advanced knowledges and bring interest to people for the first 6 weeks, and then the last weeks is about how to conduct a research. So this will be the time schedule, but it might change depending on the mental situation, but hopefully it won't change too much. So, and I enjoy the course and learning. If you don't understand after the explanation, like you can just ask me, and then you can search up on Google as we will be providing you the slides that I'm using for this lesson. And then, so yes, it's just way easier to just learn from the Google and then search up things. You learn when you search. So what are DNAs? So just basically introduction. DNA is a blueprint for our bodies, just like how different Lego blocks are created in different structures. Where DNA determines how the body is functioned and built. It's made up of nucleotides, A pairs with T, C pairs with G, phosphate, and sugar. So I think basically a lot of people know this probably. So what's RNA? It's different to DNA, which is deoxyribonucleic acid. So it's just a single strand of copy of DNA. It helps in creating proteins. All RNA are made up of nucleotides. A pairs with U, T pairs with G. So here you notice that it's different to DNA, which is A pairs with T, but this one is A pairs with U, which is uracil. So it's different. And different shapes makes them act differently. So types of DNA, mRNA, it carries the genetic instruction from DNA to ribosome. So linear shape is like the RNA is folded in linear shape, so it is called mRNA. And when it's folded into cloverleaf shape, it's called tRNA, and it reaches the codon on mRNA and brings the corresponding amino acids to the ribosome. And globular shape is rRNA, which is RNA, and forms the core of ribosome and helps catalyze the formation of peptide bonds between amino acids. And we will continue this further in the slides at the back. So proteins. Proteins are formed when peptides made of amino acid chain together to form polypeptides and later fall into shapes. They have a lot of jobs: build structure, fight infections, speed up reaction, enzymes. And yeah, if any question, just raise your hand and then ask. Central dogma is a theory stating that genetic information flows from only in one direction, from DNA to RNA to protein, or RNA directly to protein. So like this, it's just a theory of like how does it work in protein synthesis thing, and we will talk about it later as well. So DNA hosts the instruction, RNA copies and deliver them, ribosomes build proteins, and protein works for the body. So it's just what their roles, each roles are for. So how do we make the protein? Through protein synthesis. Does anyone know this? Like learn from anywhere else? Okay, no worries. We're going to learn this anyways. So there's two stages for the protein synthesis. First stage is transcription, which is from DNA to RNA in the centromere uh, theory. The two strands of the DNA helix. So the DNA helix is like this, and there's two strands, and they are bonded with weak hydrogen bonds together. So uh, is unzipped, is separated by breaking the weak hydrogen bonds between base pairs, so like A and T, C and G, and then caused by an enzyme called helicase enzyme. The enzyme RNA polymerase, which is a chemical that just does such a thing and then make mRNA, attaches to the DNA in an encoding region just before the gene. So just line up first, and then it will start copying the DNA in complementary pairs. So RNA polymerase, the enzyme that builds RNA, moves along the DNA. Free RNA nucleotides, AUCG, so the materials for making RNA floats in the shell in the cell. So they just basically pick up the materials in the cell and then just build it. Match up with the exposed DNA base here, like fitting puzzle pieces to form a new strand of mRNA. And then, so the newly formed strand of mRNA is now ready to leave the nucleus and travel to ribosome. So to recap, it's just the the DNA is unzipped by helicase, and then the RNA is uh the RNA polymerase just makes the uh complementary pairs this strand to further to modify in the next stage. So let's carry on. So the next stage is translation, RNA to protein. The mRNA strand travels through the cytoplasm, where the fluid uh, thing in the cells and attaches to the ribosome. The strand passes through the ribosome and gets in there. For every three mRNA bases, the ribosome lines up one complementary molecule, anticodon. So the free RNA is a codon, which stands for an amino acid. So because it's according to those three RNA and it's a different pair. So like this is codon and this is an anticodon. So you see U and the complementary pair is A. So this is codon, this is anticodon of tRNA. So here you can see this. So these are each amino acids that's binded to the tRNA. So of tRNA called replaces codon. mRNA codon AUG, tRNA anticodon UAG. So just flip around. A to U, U to A, G to C. Here. So like this. tRNA molecules transport specific amino acids to the ribosome, which they leave behind shortly after lining up opposite of the mRNA. Because there are three mRNA bases for each tRNA molecules. You see, three mRNA bases to one tRNA molecule, one amino acid. So we call it the triplet code. Each tRNA carries a specific amino acid that matches the anticodon. Use tRNA exit the ribosome and collect another specific amino acid, just continue the process in a cycle. So as amino acids are like material to like when it's like monomers and then when they bind it together, it will become a polymer, which is a long chain, which is polypeptide, polymer. So polypeptide is a protein, like it is then folded into protein. This is basically the thing. It's protein, but before it's folded, a chain, correct order. So that's how the protein is made from the instruction in the DNA. Does it and make sense? If it doesn't make sense, then raise your hand or something. So let's move into cellular processes. So perform specialized function to sustain life, mitosis, respiration, signaling, and transport. And these are just basically how these words are meaning in simplified. So mitosis, it makes body cells, skins, muscle, organs. One division is it becomes two identical diploid cells, 46 chromosomes in human. So they are like a pair of chromosomes in each uh, chromosome ones. So this 46 is there's 23 kinds of chromosomes. Purpose, growth, repair, and maintenance for making body cells. Meiosis makes sex cells only, so sperm or eggs, which is different to mitosis. And two divisions make four unique haploid gametes. So haploid is half, diploid is a whole. So this one is 46 and this one is only 23 because it's haploid. It only takes half of the things. Purpose, reproduction, to pass genes to the offspring. So here is cell cycle. Mitosis is the process of somatic cell division. So somatic just means the body cell, excluding the sex cells that enables growth and tissue repair. Meiosis, a special type of cell division that makes sperm and egg cells, so the haploid gametes. Um, and do you think I should slow down a bit or but I'll just keep on this pace probably. Like interphase, the DNA in chromosomes copies itself ready for mitosis. So if you learn like GC in school probably there will be like a table, a circle table, a chart, which is uh like S and then interphase. So interphase is not included in mitosis, but it's part of the processes that leads to mitosis. It's before mitosis. So interphase, the DNA is copy itself ready for mitosis. So it doubles the material for the mitosis to split apart into two identical cells with the same amount of materials inside. Prophase, chromatids condense into chromosomes to become very more visible. The membrane around the nucleus disappeared. The membrane is also called the nucleus envelope. But yeah, but and then chromatids is basically a chromosomes is the whole structure of the DNA thing. And then chromatids is basically the identical size uh of the chromosome, so half of the. So this is 46 chromosomes. Like if this is up here and then 46 chromosomes, and 46 chromosomes. And this is also 46 chromosomes, but it has, because of interphase, it's double the amount. So it's now 92 chromatids. So I hope it makes sense. And then metaphase, chromosomes and their copy lines up in the middle of the cell. So like this, lines up in the middle of the cell. So this doubled lineups. Anaphase, chromosomes and their copies are put to different ends of the cell. So it's pulled by the spindle fiber made of centrioles. So there's a string in two ends, and then it just drags it across to opposite ends of the cell, and then it's divided. So interphase, prophase, metaphase, anaphase, put to different ends. Telophase, new membranes form around the chromosomes at each end of the cell. So here is forming uh, the cell membrane and then trying to separate it into two cells. Cytokinesis, the cell membrane pinches in and eventually divide into two daughter cells. So here is the cell membrane has completely divided into two. Well, in that phase, yeah, just meiosis is similar processes to mitosis, but it has two rounds of stages. So prophase chromatids condense into chromosome and the membrane around the nucleus disappears. The chromosomes from parent cross over and mixed. As we have two chromosomes for the 23 kind. So how it is mixed is basically just crossover. The two X's is crossover in different proportion, and which we call it as bivalent, which means two, a pair of them, and then it is turned into this mixed, but it can be different proportion. It doesn't have to be like completely 50-50. And then metaphase, chromosomes and their copies line up in the middle of the cell. Anaphase, chromosomes and their copies are put to different ends of the cell, so similar to mitosis. Telophase, new membranes form around the chromosome, same thing. Cells split into two half-blood cells, different to mitosis, which is diploid cell, and membrane closes, so it's similar to mitosis. And then meiosis two, this is meiosis one. So you probably see prophase one and then metaphase one in meiosis one. So there's interphase, prophase one, metaphase one, anaphase one, telophase one, and then cytokinesis. Uh, and then meiosis two, and now it's prophase two. So here is interphase, only one in the whole thing. So because it has double the amount of of the chromosomes from than mitosis, in mitosis there's only a pair, whereas this one there's just four of them because both the both of the parent cell materials are mixed. So there's four. And then now it's doubled and then it's split into two cells with double amounts of the materials than the mitosis. So that is meiosis one, and this is meiosis two. So this one chromatins condenses into chromosomes, and the membrane around the nucleus disappears. It should be chromatized, I think. Metaphase, chromosomes and their copies line up in the middle of the cell. And then anaphase, two homologous chromosomes are separated and put to different ends of the cell. Telophase, nail membranes form around the chromosomes at each end of the cell. Cells split into four unique haploid cells. So like this at the diagram. Homologous split. Now is the more like the most difficult part of the session, set of cellular respiration. Uh, Cellular respiration is a metabolic pathway that uses glucose to produce adenosine triphosphate, so ATP, an organic compound that releases energy by breaking its high energy phosphate bonds in hydrolysis water. So hydrolysis is basically you add water in there and then the bond breaks because yeah because it's the water has substituted the phosphate bonds and then make them separate as they are completed. And you might see condensation as well, which is two of the two of the uh, polymers, two of the monomers, and they bind it together, and water is produced after that. So when you add water in there, and the water is used by the reactant and become a whole product, and in condensation is uh, a whole product, and then split into two with a waste product as water. So, uh, yeah. So one molecule of glucose can produce a net of 30 to 32 ATP. So yeah, glucose is so in mitosis and in mitochondria is glucose is made into energy. So here it explains why. So glucose is producing the ATP in the mitochondria. So here is the respiration. Basically, you probably know glucose, oxygen, just the respiration formula, CO2, H2O, and the ATP, which is will be turned into energy in mitochondria and release energy. So it can be used for cell division to maintain constant condition in cells and the body, homeostasis. So homeostasis is maintaining constant condition like temperature or pH, acidic thing in the body to move molecules against concentration gradient in active transport. So like uh active transport uses the energy to move something from a lower concentration to a higher concentration. It doesn't isn't uh used in diffusion because diffusion you can only use for high concentration to low concentration. For the transmission of nerves impulses, so another example for what the energy from respiration is used for. So there are three main steps for cellular respiration: glycolysis, the citric acid, and the Krebs cycles, and the electron transport chains. So glycolysis, TCA or Krebs cycles 1, 2, and then electron transport chain 3. So glycolysis, the first step is breaking down glucose, which is 6 carbon, into two pyruvate molecules, 3 carbons each. So glucose is 6C6H12O6. This is just C3 something. And then happening in the cytoplasm. Key points: when glucose is turned into 2 pyruvate plus 2 NADH plus 2 ATP. Doesn't need oxygen. So 2 pyruvate transported into mitochondria matrix and oxidized into 2 is CoA, carbon dioxide, and two NADH. So it just continues. And NADH is basically just a coenzyme that transfers electrons. And coenzyme is just basically supporting the enzyme for working. And then pyruvate and acetyl CoA, the reason to break down from glucose to these two is because they are smaller, so it's easier to process than a big molecule. So Krebs cycle, citric acid cycle, yeah, aerobic process occur in mitochondria matrix. So a cycle where two acyl CoA will enter, carbon dioxide is released and will produce two ATP, six NADH, and two FADH, which is similar to NADH and both transfer electrons. So you just need to do those two things. Yeah. Like similar, but a little bit different. So electron transport chain and chemiosmosis. So chemiosmosis is basically just ions going from transferring over a membrane and then from a high electrochemical gradient to a low electrochemical gradient. And then it's also an aerobic process and occur in the inner mitochondrial membrane, a coordinated series of redox reactions that extract energy from high energy electrons donated by NADH and FADH2. Use this energy to pump protons across the inner mitochondrial membrane into the intermembrane space, create both a chemical gradient and an electrical gradient. So electrochemical gradient, yeah, here. At the end of the electron transport chain, the final electron acceptor, O2, combines with the protons to produce water, as proton is just H, is just hydrogen and the exit. Meanwhile, ATP synthesis uses the movement of protons back into the mitochondrial matrix for ATP synthesis. So the here water is just hydrolysis, which we talked about earlier. So here is how it works. So basically, there's a router inside the mitochondria. It's pumped energy from electron transfer causes protein shapes changes in complex 1, 3, and 4, so complexes, molecules. These changes forces H plus, so proton, from the matrix intermembrane space like a mechanical pump. How ATP is created, the proton spins the F0 rotor in 100 times a second. Each full rotation produces 3 ATP in F1 factory per 10 H plus proton. So here, so here is the router. It's a biological router, and then the protons from here is going into the intermembrane space, and then it produces uh the kinetic energy, makes the makes it producing the free ATP in F1 factory per 10 protons. So you make ATP at the first step of the uh, of the like the glycolysis of the cellular respiration. And then 10, and then this one you also create the ATPs. And then it adds up to 30 to 32 ATP per glucose. Wait, my laptop is charging. Wait for a second. Yeah, let's carry on. So here is the diagram for for this. So glucose, ATP, and AT plus. So here it makes ADP and then makes ADP, ADP, and then all the steps the ATP produce add up together is 30 to 32 ADP per glucose. So here is the last things, basics of genome editing, a technology that allows scientists to precisely alter DNA sequences in a living organism. It helps fix mutation and traits or study gene functions. Methods CRISPR-Cas9, which you probably know that is like most popular, cheap, versatile, Talin, high precision, complex design, and a zinc finger nucleus. First generation, less flexible. So this is like the old methods, and this might still be used. And then this is the most commonly used and new technology. Example, in current studies, researchers uses CRISPR-Cas9 to modify genes like GN1A and IPA1, which control grain size, number, and growth speed in rice, solving global food shortage, and also make golden rice, which helps beta carotene which convert to vitamin A in body deficiency in poor countries. So this is one of the example on how they use the genetic modification to create something that helps world hunger. So these are some of the ethical questions. So designer baby's risk. In every technology, you need to understand the risk and consider the problems. And editing embryo for trace for trace like intelligence or looks raises moral concerns. Unequal access, expensive treatments could widen the gap between rich and poor. Unintended harm of target edits might cause neural disease or side effects. Ecological damage, modified organisms could disrupt ecosystems. No consent for future generations. Edited DNA passes to offsprings without their approval. Potential misuse could be weaponized to create harmful bioengineered pathogens. But these are all the ethical issues people have considered for this revolutionary uh, technology, and there was also more, but it's just enough. So thank you. It's very good to see you guys. And then the the whole lesson is complete. And then is there any questions? There's still like 3 minutes left in the meeting. If not, it's fine. Okay. Yeah, thank you, Amma. So yeah, thank you. Thank you, Laura, for the comment. And so I'll just send the PowerPoints to you guys, and then you can revise it or check it on Google if you have any questions or stuff. So yeah, that's it. I will see you guys probably afterwards in I don't know which week, but yeah, I'll see you. Bye. We can go now. See you. So no one has questions, so then I'll end it. Bye.